begin by acknowledging the traditional owners on the lands from which we're gathering today, and respectfully acknowledge their elders, past, present, and emerging, as well as any members of the ATSI community gathered amongst us here this afternoon. By way of brief background, the MPCCC Precision Cancer Program is a community of researchers, health professionals, and academics from across Victoria with an interest in precision medicine and particularly in precision applications of precision cancer. We're working and have worked indeed for the last few years to establish better access to molecular sequencing for cancer patients across Victoria and elsewhere as requested. And we also host several molecular tumour boards that support the interpretation and clinical application of the molecular data that's generated from from the sequencing of patients' tumours. These seminars that we're hosting today are another important part of the program, which also seeks to undertake workforce development and education activities such as these. And these seminars certainly help to share the latest research activity across our own partnership, across our state, but also nationally and internationally, and to communicate new ideas to our community around approaches, some cutting edge approaches to clinical care. So along this line, it's my great pleasure to welcome one of, I think, our best, most innovative and creative scientists, Dr. Alex Swarbrick, as our guest speaker today. I've known Alex for some years through collaborations in various fora, and he really is one of, I think, the most exciting scientists that we're lucky to host in this country. Alex is a Senior Principal Research Fellow and co-leader of the Cancer Ecosystems Program at the Garvan Institute in Sydney. He's a conjoint Associate Professor at the University of New South Wales and an NHMRC Senior Research Fellow. He completed his PhD at UNSW and a postdoc fellowship in the Bishop Lab at UCSF. His lab applies cellular genomics to breast and prostate cancers and melanomas, seeking to gain system-level insights into the etiology of this disease, the way it progresses, and interacts with the environment, ultimately all towards developing novel treatments. It's such a pleasure to welcome you, Alex, and we really look forward to your talk. There will be some questions, some minutes for, or some time for questions at the end of Alex's presentation, and so we'll kind of reserve them for then. Certainly if they come to you during his talk, feel free to pop them in the chat, and then I'll kind of batch them up for moderation at the end of the talk. So for now, just please mute your microphones, turn off your videos so that we can only see Alex and not the rest of you, and generally minimize disruptions. And I'll hand over to Alex.
Thank you, Mark. Hi, everyone. And thank you for that really lovely introduction. That was very generous of you. Never one to shy away from hyperbole, but I'll take what you'll give me. And thank you, everyone, for joining today. It's really nice to see friends and colleagues and a bunch of people I've never met that I hope I can eventually meet. So today, I want to talk to you about the work we're doing studying solid tumours as cellular ecosystems. I'll really focus on our work in breast cancer because I think it exemplifies the kind of thinking we're doing, but I think there are many elements of what we're finding in breast cancer that will apply to other solid disease systems. So just my disclosures to begin. So just briefly, I want to cover the kind of clinical challenges of what we're trying to address. Some of what I'll go over today is quite technical, sometimes mechanistic, but ultimately my lab is a translational lab. We want to try to shift the dial on treatment like I'm sure many of you do. And these are some of the problems that we're interested in. You know, the advent of kind of cancer genomics has absolutely revolutionized the way we study the disease. There's no denying it. and it's certainly changing cancer treatment. But perhaps not surprisingly, it's not the entirety of the answer in itself. And so the survival rates for a lot of solid cancers are still way too low. A lot of patients receive non-personalized treatment, and often that treatment is futile. And so really we're interested in looking at some of the most prevalent solid tumors, breast cancer, pancreatic cancer, prostate cancer, with collaborators to try to understand why people fail to respond to standard of care therapy, and then what we can do about it. And the place where we're looking is in the kind of using a cellular model for disease rather than not so much a genomic model for disease. So this is what I want to try to introduce before I get onto my data. So, I don't think you need to be a pathologist to look at the H&E section of a breast cancer on the left here to see the kind of wild diversity of cell types that are present, everything from cancer cells to invading immune cells. You see these huge rivers of collagen and extracellular matrix here. And we know now that obviously cancers are a really diverse collection of cells, and that there's huge diversity within these populations as well, within the immune infiltrates, within the stromal populations, and within the cancer cells as well. But mostly through technical limitations, as well as some conceptual kind of bottlenecks, we still don't really understand this kind of cellular environment of solid cancers very well at all. And so this is the space in which we work. And so that's obviously kind of a big space. And so we try to break it down into areas of research that we think are approachable. And this is the way we think about the problem. The first is really a problem of taxonomy, in a way, to understand the individual parts, kind of a reductionist strategy. What are the cell types that comprise tumors? What are the cell types that we need to think about and worry about? And what are the kind of levers we can pull to change their behavior, to activate them, to ablate them, whatever it is we might want to achieve. We then want to think about tumors as tissues, because we know that cancers aren't just a sack of cells rattling around like marbles. They form structures just like cells do in healthy, non-disease tissues. And this is an area where I think there's a huge amount to learn, and a really exciting area, and one in which new changes in technology are allowing us to start to kind of break open what were previously black boxes. And I'm going to talk a little bit about this. And then finally, this observation that if we look at the cellular constituents of patients, they can vary wildly. We don't think of this very much, particularly not clinically, but we do in some senses. So for example, we talk about an inflamed tumor or a hot tumor versus a desert. And in that setting, we're talking about the TILs, the tumor infiltrating lymphocytes. But of course, there are many other cell types in there as well. And so we're really keen to understand the forces that govern kind of cellular composition in a patient's tumor, and what the relevance of that is to their management. So just to kind of distill this down to, I guess, some kind of actionable points, here's a prototypic cell in two different environments. And as it moves into this new environment, it changes phenotype. And this could be a cancer cell shifting from a drug sensitive to a resistant state. It could be a stromal cell shifting from like a tumor suppressive state to one that promotes tumor progression. Or it could be an immune cell going from, for example, an effector state to a dysfunctional state. And the kind of forces that are feeding into this decision or this event are many. And these are some of the ones we're interested in. The first, and one that we're very focused on, is the cellular neighborhood, how one cell impacts an adjacent cell to change its state. Another that colleagues at Garvan work on, such as Tom Cox, is understanding the factors outside the cell, like the ECM, and the role that they play in coordinating cellular kind of differentiation and fate decisions. There's emerging evidence that somehow, and through mechanisms we don't very well understand, the different epigenome or genome of the cancer cells can then dictate the resulting phenotype of the microenvironment around it. And of course, we know that drug treatment drives a huge number of kind of state transitions across all the major lineages that we see in a solid tumor. And so really, our aim is to kind of firstly, as I said, understand the individual cell states, and then start to understand the role that factors like these play in determining cellular phenotype, and how we can end up using that to make decisions about treatment and perhaps pushing tumours into a better outcome or more treatable kind of state. I want to give you an example of that because that's a bit kind of esoteric here, and I'll use some of our own work as an example, but there are many examples now in the literature. Some years ago, we worked on triple-negative breast cancer as a very aggressive subset of breast cancer, and we showed that in a subset of cases, these patients activate expression of an extracellular ligand called hedgehog. And this hedgehog binds to the receptor patched. And we showed actually that the key target for this hedgehog patch signaling were adjacent fibroblasts or cancer-associated fibroblasts or CAFs. And when tumor cells signal to the CAFs, they activate the CAFs to firstly remodel this fibrillar collagen. And we know that changes in ECM structure can greatly impact cancer behavior, but also secrete factors like fibroblast growth factors that can also activate cancer cells. And the net effect was to drive the cancer cells into an aggressive drug-resistant state through this kind of circle of activation. And we were lucky in that there were small molecule inhibitors available to smoothen for a very different indication for brain cancers and skin cancers where you actually get genomic events in smoothened. So a fundamentally different biology, but we were able to repurpose the drug, and through a series of preclinical models and then eventually into clinical trials, we could show that if we take patients who have failed on conventional therapy, so this will be lines of chemotherapy in this setting. These are patients with metastatic triple-negative breast cancer. They're really in trouble. You know, they're failing to respond to the chemotherapies that are really our major weapon for them. And if we treat these women with a combination of docetaxel, so a cancer cell targeted agent with a smoothen inhibitor that we think will be targeting the fibroblasts, we're able to see clinical benefit in 3 of these patients. And in fact, one of these patients that I'm showing you here, their metastatic lesions became undetectable following treatment. Now, this, of course, is a phase 1 trial, very small patient numbers, we didn't have a control arm, so there are a lot of reasons why we might have made this observation. But I think it's consistent with the ideas and kind of really excited us to try to go further to examine this area of biology. And, you know, in a way, we got lucky in finding this pathway. And so we wanted to take a more informed approach to discovering these kind of signals that go on within tumor microenvironments. And we saw the emergence of cellular genomics as an opportunity to do this. And so, you know, at least at the time, cellular genomics was not compatible with the large majority of the tissue biobanks that we have available. It generally didn't work on formalin-fixed paraffin-embedded tissue. And so we set about setting up some fresh tissue collection programs across Sydney, collecting from patients who are very, very kindly donated excess tissue to us, whether that be at first emergence of disease, at metastatic relapse, and also I ran a national rapid autopsy program to capture participants who would donate their bodies to us after death. This was called the Brocade program that let us study kind of very frankly advanced metastatic disease. Okay, and then we wanted to apply cellular genomics to this. And at the core of this is always single-cell RNA sequencing for us, at least up until now. It tells you so much about the phenotype of a cell and of a tumor. And essentially, you're counting transcripts from thousands of cells that you sample from every tumor. But because these are clinical specimens and we often get one shot at it, we wanted to try to add as many dimensions of data as we could so that we could learn as much as possible as we could. So the first thing we did was to develop a method called RAGE-seq. That stands for receptors and gene expression. And this method was developed by Gami in the lab. And the reason we did this was to try to get together with the gene expression at cellular resolution to be able to sequence T cell receptors and B cell receptors, which is, of course, of great interest if you're studying tumor immunology. And so this is a really kind of neat method for doing that. You get these kind of 2 data sets in parallel. Now, of course, as I mentioned, tissue structure is critically important. And so we've been working with tissue-based assays, and I'm going to show you some data there eventually. And then finally, we adopted a method that I think is really great called CytSeq. And essentially, this method uses barcoded antibodies to detect antigens on the surface of cells, just like you do with flow cytometry, but instead of using a fluorophore here, we're attaching a barcode that we measure through sequencing. And the beauty of this is that it gives you, again, in parallel single-cell RNA sequencing, but because it's a FACS-like method, it links us back to the canon of knowledge in immunology, which is based on FACS. So we can kind of link these new technologies back to this huge literature based on flow cytometry. It's very sensitive, and often we can measure the cognate protein much more readily than we can measure the associated RNA transcript. And because we don't have to worry about interference between fluorophores, which you do with flow cytometry, there's actually no theoretical limit on multiplexing. And in fact, we're just starting an experiment now with a 350 antibody panel, which is pretty wild, I think, when you think about the power of that for immunophenotyping. And finally, if we're thinking translationally, we need to know the expression of proteins on the surface of cells. We need to know the expression of the receptor or the ligand that we're considering targeting with therapeutic intent. Just to show you, give you a little insight into the CytSeq panel that we have been using to date. It's 157 targets, really kind of spread across innate and adaptive immunity, as well as a handful of targets on cancer cells and stromal populations. And so what I'll show you now is some data from what's becoming a really big project in the lab, which we call the Breast Cancer Atlas. And this is based on these cohorts that I described to you of localized as well as metastatic disease. And we want to kind of generate multi-layered data from these patients so that we can start to link findings, so we can understand how genomes are regulating cell types, for example, or how cell types impact on patient outcomes. And so using a diversity of methods, we're analyzing kind of cell types and states and the molecular features. We've just started a program to do germline and somatic genomics on this cohort. Here, we're generating spatial data with our colleagues. We're also profiling the matrix structure of all these cases, as well as performing bulk proteomics with the team at ProCan at Westmead Hospital, and then really importantly, linking that back with our clinical colleagues to the histopathology and clinical outcomes for this cohort. So I'll show you some data now from a recent publication. It's really the first snapshot of this cohort where we studied 26 of these localized breast cancers. We've just generated data for 260 cancers, so an order of magnitude bigger, but it's going to be a little while before I'm able to show you that data. But I think there are some interesting lessons from this study. What I'm not going to do is kind of walk you through the really descriptive stuff, because you can go see that in the paper, and I think that can get a little dry. But what I want to do instead is to start to focus in on a couple of areas that we think are interesting and where we're trying to pull out biological insights and insights of translational relevance. So the first one is in immune cells. So, checkpoint immunotherapies have been transformative for many cancer patients, and they're particularly effective in certain diseases like melanoma, for example, where Mark works. But in breast cancer, the results have been fairly disappointing, unfortunately. A relatively low proportion of patients will respond to checkpoint immunotherapy, particularly have kind of durable responses to it. And so one of the questions we really want to understand is, why is that? What are the determinants of it? And how can we maybe modify the strategies for immunotherapy in breast cancer patients to make them more effective? And so if we look now at the kind of T cells and associated cells like ILCs, which is where you might expect some of the action to be happening, we can see a number of populations that are of relevance here. We've got proliferating T cells, we've got exhausted subsets. Cluster 6 here are interferon-activated T cells. And then I'll draw your attention to two other populations, which are these CD8 positive T cells. So these are probably TRMs. And over here, we've got this really interesting and quite substantial subpopulation of CD4 cells that look, for all intents and purposes, like a T follicular helper cell. And I'll come back to these cells in a minute. Interestingly, some of these populations are asymmetrically distributed between subtypes of disease, particularly these populations I've shown you here being enriched in triple-negative breast cancers. And this is where we see most of the signal from checkpoint immunotherapy, which this may be part of the explanation, a pure kind of stoichiometry that there are more of these cells and able to respond. So using CytSeq, we wanted to try to go deeper on phenotyping these cells. And one way we can do that is to cluster the cells from the single cell RNA sequencing and then project the matching protein expression onto these cells. And this can be really helpful actually in distinguishing between populations. I'm showing you an example here, these two populations, which both look very much like NK cells, but you can see that only one of them expresses T cell receptors. So these are actually NKT cells, whereas these are NK cells, for example. And that's difficult to discern using the RNA data alone. We can also map all these kind of checkpoint ligands and receptors that are in our panel onto these populations. And interestingly, one of the points here is that these TFH cells, these CD4 T follicular helper cells that I pointed out, they're really blazing for PD-1, which we know from immunology, but it's interesting to see this also apparent in a solid tumor, that one of the biggest sinks of PD-1, which of course is the major target for checkpoint immunotherapies, is actually these CD4 T follicular helper cells. And I want to dive into that in a moment. Really, as an aside, I couldn't help but show this as a pure kind of technical development, some really new unpublished data led by Gami, Holly, and Marlon, a collaborator from 10x Genomics, where we asked, well, we can do CytSeq on cell suspensions and use single cell to measure it. Can we apply this to tissues? And so these guys kind of hacked the Visium spatial transcriptomics system to allow us to stain those tissues with barcoded antibodies in essentially the same manner, and then read that out through sequencing. And so I'm showing you just an example here of actually generating 3 different domains of data on the same target. This is immunofluorescence, classical indirect immunofluorescence for EpCAM, showing you the high expression of EpCAM in this piece of solid tumor here. This matches the expression of EpCAM protein from the barcoded antibodies, and it matches the expression of the EpCAM transcript that we get from whole transcriptome sequencing across all of these spots. So this is pretty cool. And so we can now, in a spatially resolved way, measure this. Gami, being the funny guy he is, decided to call this SpiteSeq, just for the lols, more than anything. But he's also taken his RageSeq method and managed to hack that to work on tissue as well. And so what I'm showing you here now is in actually the same case, though the pieces are in a different orientation, is our ability to also now map B cell and T cell receptor clones spatially. So we can now get these kind of 4 dimensions of data from this one piece of tissue, which I think is going to be really exciting in deeply immunophenotyping lymphocytes and the way that tissue location affects their behavior. And I'm happy to talk more about this later. I know I'm really kind of skimming across the top of this. Okay, so back to the single cell CytSeq. I showed you here that we can use a simple method of just kind of mapping the protein data on top of the RNA clustering, and that's useful. But what Gami really wanted to do was to ask, can we integrate the RNA and protein data and use that to get higher resolution insights into cellular phenotypes? And the answer is yes, we can. And so the details here don't matter. What I want to show you is that when we cluster on RNA alone, these are the clusters of cells that we get from breast cancers. If we cluster on the protein data alone, we get a smaller number, probably because there are less measurements. But when we integrate them, we get this massive expansion in our ability to resolve populations. And many of these are populations that immunologists have previously resolved using really painstaking targeted flow cytometry methods. So we think these are real populations, and we can finally see them within this single cell data. And as an aside, one of the places where this is most useful is when you're studying cells with low transcriptional activity, like a resting lymphocyte. There's simply not a lot of RNA there to measure to let you understand who it is and what it's doing. And so the addition of this protein data gives us another dimension that lets us be confident about the identity of these cells. The second point to make, of course, is there are certain antigens that you simply can't measure from the RNA. And for the immunologists, an example would be CD45 RO, which tells you about antigen exposure of cells. And this is the consequence of a splicing event. You can only measure it using protein, not short read RNA data. All right, so I mentioned before these T follicular helper cells. We're seeing a lot of them in breast cancers and we're seeing very high expression of PD-1 expression on these cells. And so we're interested to ask, what are they doing in breast cancers? Because the role of T follicular helper cells classically in immunology is in secondary lymphoid organs like the lymph node, where they really serve to help B cells and T cells in their response to antigens. So that's why they're called follicular helper cells. They're in the follicles, helping kind of generate useful responses, including in response to COVID vaccines, which I'm showing you here. So when GAMI looks closely and using some methods where you can infer differentiation of cells, he's now zooming in on these T follicular helper cells. And actually what he sees is it appears there are two trajectories that these cells can go down in the kind of decisions they make. The first is a conventional path, which leads you into what really, for all intents and purposes, looks like one of these cells that you expect to find in a lymph node. And what they're doing in breast cancers is interesting. We're not sure, but we have some clues. The second is a phenotype that you will never see in a lymph node. In fact, that you'll never see in physiology at all, which is in fact a population of these CD4 TFH cells that look, for all intents and purposes, exhausted, like an exhausted CD8 cell. And interestingly, it's these exhausted CD4 cells that correlate with prognosis if you stratify breast cancers using signatures of them. So we were pretty interested to look more closely at this. We've got now a novel population that you don't see in physiology, but which is quite abundant within breast cancers associating with prognosis. And in fact, when GAMI takes all lymphocytes and simply clusters them based on their gene expression or based on the pathways that they have highly overrepresented, either way you look at it, what he sees is these exhausted TFH cells clustering right next to exhausted CD8 T cells. So they are their most similar cell type. Despite being from the two lineages of T cells, they've kind of come together in their phenotype, which is really fascinating. So we wanted to use spatially resolved transcriptomics to test the hypothesis that this is a function of a unique cancer microenvironment. Just like I said at the beginning, perhaps there's a unique environment that's driving these TFH cells into this weird phenotype that we wouldn't normally see in a healthy tissue. And so just to kind of give you a little more background on this method that I briefly touched on before, it's essentially a microarray method where the manufacturers have spotted 5,000 spots across a glass slide covered with capture probes that can capture mRNA transcripts and then attach a location barcode to them followed by sequencing. So when you collect these together, sequence them, you can map reads back to each of these spots. And you can see here in this example, this three very different pieces of tissue, this one full of normal mammary ducts, this one full of cancer and TILs, and this one full of more cancer, that if we map the signatures from our single cell sequencing to them, they map very nicely to the pathology that we expect. And so the point is that we believe that spatial transcriptomics very nicely matches pathology and is kind of reading out truth to us. And here's another example. We can start to look at higher resolution, for example, mapping subsets of T cells. We can see where the CD4 T cells reside in this piece here. It's in this aggregation of TILs that the pathologist has previously marked up. Whereas interestingly, the CD8 T cells are more concentrated in this piece of tissue here. Okay, so now when GAMI dives into a higher resolution now and starts to ask, well, where are these cells? Here's some of the data on the left from a single case. You can see that B cells are clustered in these aggregates here, which we think may actually be tertiary lymphoid structures or TLSs together with dendritic cells. And that in fact, again, these conventional TFH cells are found in the same place. So probably doing what they're meant to do in a lymph node. Whereas these exhausted TFH cells, you see many of them scattered through the tumor here. And this is kind of summarized in this heat map across a series of cases now, where these conventional TFH cells are mostly found in immune aggregates, whereas the exhausted TFH cells are mostly found in invasive cancer regions. So supporting the idea that these two populations that come from the one progenitor end up in different environments, and perhaps that's what's driving them into these unique states. GAMI can also show that in addition to being near cancer cells, these exhausted TFH cells are also associated with macrophages compared to the conventional ones, which are not. And in some kind of cool analysis where we can predict ligand receptor interactions. So in other words, what kind of communication might be going on here. This analysis argues that these exhausted TFH cells produce lots of colony stimulating factor, which is a classic mitogen for macrophages. And you can see it signaling to a range of these monocytes and macrophages. So a series of lines of evidence that these cells are interacting with really different cells, and maybe that's what's changing their phenotype. Now, at about the same time we were finding these, a paper came out very recently coming from a very different direction, where they asked in solid tumors, starting in melanoma, whether they could find neoantigen-specific CD4 T cells. And in fact, they do. And the subset of these CD4 T cells that they find them in are CXCL13 high cells, which is the same phenotype as we see in these T follicular helper cells. So here's a completely different study, but again, suggesting that these cells are not only adopting this unique exhausted phenotype, but maybe that's because they actually recognize tumor antigens. So they're being chronically exposed to tumor antigens. And that might be why they adopt this phenotype. So suddenly this is looking like a really interesting population. And one thing that makes us particularly interested in them is if we look in data sets where patients have received checkpoint immunotherapy and their response has been tracked. And so here, if we take these patients, stratify them into patients who responded versus didn't respond, and we ask which cellular populations at baseline associated with response to checkpoint therapy, you can see that top of this list are these exhausted TFH cells, which are at about a 7-fold higher frequency in responders, in people who went on to respond, than in those who didn't. In fact, higher than exhausted T cells, which are the presumptive target of this checkpoint therapy. So here we have a PD-1 positive population that we think is recognizing tumor antigens and which is predicting response to PD-1. All right, so that's, I think, a vignette where it kind of, I hope, showing you how we can go from these kind of broad descriptive studies to start to find populations that might be of clinical or translational relevance. So I want to switch now to a second population. As I mentioned, we try to take a systems level view of tumors. And so I want to walk you through some of our data from stromal cells, and I'm going to focus down on some of the work where we're studying the interaction of stromal cells with immune cells. So it might all kind of come back together. So if we now take the data from our breast cancer atlas, subcluster it just for stromal cells, we can see they fall out into three major populations, which we might have predicted: endothelial cells, fibroblasts, and what we call perivascular-like cells, which have kind of markers of smooth muscle cells and pericytes and so on. And so we can see that each of these is kind of a diffuse blob. And the hypothesis that we had was that, in fact, this marks different stages of differentiation within these lineages that don't interchange. And so to test that, we can again use this method where we can infer differentiation lineages. And I'll just show you the data from fibroblasts here, though we've done the analysis for endothelial cells and these perivascular cells as well. And so when we do this with fibroblasts, it actually shows indeed that there are at least two end states, but potentially some intermediate states, these kind of undifferentiated cells that actually look like resemble a mesenchymal stem cell and also have gene expression features of many kind of inflammatory markers. And this algorithm predicts that they are the progenitor that differentiates into a classical myofibroblast. This is your conventional collagen depositing fibroblast that we think about. Using CytSeq, this was actually really useful. Even though the panel was designed for immune cells, many of these markers lit up the stromal cells. And so we were able to derive a number of markers for each of these states. And what that lets us do then is correlative science on these specimens. One of the things we can do is now do detailed immunofluorescence studies where we can use markers to look at where these cells are. And one of the populations we were interested in was these perivascular-like cells. The assumption was that these were pericytes that would be blood vessel associated. So using an endothelial marker here, CD31, and a PVL marker, CD146 or MCAM, you can see that indeed these cells are definitely associated with these blood vessels here. But there are also a lot of PVLs that are not associated with blood vessels. Sorry. And what this argues to us is that, in fact, these cells are probably delaminating from blood vessels and migrating out into tumors. They look just like a fibroblast, and in fact, they share certain markers with fibroblasts. And these PVLs are probably being confused with fibroblasts previously in the literature. But our gene expression data would argue they're very different to a fibroblast. One of the pieces of evidence I want to show you to make that point is some work that we did with Tom Cox and Paul Timpson, where we purified these stromal populations, put them into culture, and then looked at their ability to deposit and remodel collagen. And you can see these myofibroblasts do what we would expect. They dump tons of collagen into these assays, and they polarize it into these beautiful sheets. In comparison, these inflammatory CAFs are very poor at doing this, and it's a really disordered collagen when they do that. And finally, these perivascular-like cells produce hardly any collagen at all. Again, arguing they're not a fibroblast, they're a different cell population. Okay, so as I mentioned, from the phenotype of these ICAFs, they expressed a lot of molecules that we associate with immune activity and actually immune suppression. And so we're interested to ask whether there's further evidence we can get that ICAFs associate with immune populations. And so again, we turn to spatial transcriptomics, and we're able to localize inflammatory CAFs and myofibroblastic CAFs in tissue. One of the things that surprised us actually is that these cells, again, occupy entirely different domains within the tumor. Even though the ICAFs are the progenitor for MyCAFs, they move into totally different spaces. You can see that they're almost inversely proportional here. And what's interesting is that these ICAFs overlap with CD4 and CD8 cells. And I'll show you some quantitation which helps make that clearer. Here I'm showing you now 14 breast cancer cases, and the color of these squares shows the strength of the interaction. And I'm showing you, in this case, the interaction between ICAFs and subsets of lymphocytes. It's all red, which argues these cells are frequently found together, whereas MyCAFs, it's generally blue, arguing that they're not. And so again, this is a specific phenotype of the ICAFs. And in fact, we can use this data now together with the ligand receptor predictions to drill in and say, well, can we predict the molecules that ICAFs might be signaling with to lymphocytes? And indeed, we can do this by filtering with our single cell data for ligands that are unique to ICAFs, receptors that are unique to CD8 T cells, and then using the spatial data saying, when do we ever find these together in one place? And this ends up being the kind of list of candidates that we get. And the nice thing is we're getting hits that we would expect to find, known interactors between fibroblasts and T cells, but also novel things. Okay, so that's a bit of a blast through these first kind of sections here, starting to look at taxonomies and communities. And I want to finish on this idea of the kind of highest level view, which is cellular composition or stoichiometry, or what we call ecotype. And so if we want to explore this, the problem is that we don't have a lot of cases. We've so far got 26 breast cancer cases here. While this is the biggest kind of single cell data set in breast cancer available, it's still a very small number of cases, particularly if we want to associate this with outcomes. And so what we can do is use a computational trick where we use a training set from single cell data to get signatures of every cell type. And then we use that in very large published data sets of bulk RNA sequencing to deconvolute them and predict the proportion of cells in every one of these cases. We then cluster patients based on the predicted proportion of cells, if that makes sense, and then look at the features of each of those different groups. So that's what we call an ecotype, a group of patients with the same cellular composition in their tumor. And so this is what this looks like. In breast cancer, it looks like we've got something like 9 ecotypes when we cluster patients. So each one of these groups is a series of patients. This is a data set of 2,000 breast cancer patients with bulk RNA sequencing. Every column is a patient. Every row is a cell type. And each of these groups is an ecotype. So just to kind of let you in on one of these here, I'll show you, for example, this group here, Ecotype 4. This is driven by high levels of particular myeloid subsets, differentiated PVLs, exhausted CD8 T cells, and so on. So the first thing this tells us is that, intriguingly, these cells always go together. And that's something we don't yet understand, but it argues that somehow when this subset of myeloid cells are present, you usually also have all these other cell types. So we're already getting hints of how cells might form into kind of functional units that co-occur across patients. The second thing that's really interesting is that every one of these ecotypes is driven by cells from every lineage. So this isn't just a surrogate for a particular cancer phenotype or a hot, like an inflamed lymphocyte high tumor. These ecotypes are all driven by immune cells, cancer cells, and stromal cells. What's interesting is that they're not simple surrogates of the current methods we have to stratify breast cancers. So one of these methods is called intrinsic subtyping. And you can see that while some ecotypes are heavily skewed towards a particular intrinsic subtype, like basal's here, many of them are a mixture. So it's not a simple surrogate for that. And similarly, this other method known as integrative clusters. Again, most of our ecotypes are a big mishmash of different integrated clusters. So it suggests it's telling us something different. And what's kind of interesting is that these ecotypes have markedly different prognosis. You can see here. And in fact, one of these, here's an example comparing ecotype 4 to ecotype 7. These actually have very similar features in terms of the subtypes of patients that went into them, but fundamentally different outcomes. So we think this is potentially a new way to think about stratifying disease, that this is in a kind of unselected cohort. All we have is long-term survival as an outcome. What we're very interested to do now is to ask whether ecotypes predict response to treatment. And you might expect that one place we should go looking is response to checkpoint immunotherapy. The other place that we want to look is in dynamic systems. So start to understand how these things matter through treatment. And so I just wanted to touch briefly on a study that we're kicking off now to show you where I think this field is going, is into longitudinal sampling of patients. And so working with Meng Hui and Sandra and a couple of surgeons, Sanjay and Cindy at Chris O'Brien Lifehouse, we've assembled a cohort of patients who are receiving neoadjuvant chemotherapy, which is really a backbone of treatment for breast cancers. And a lot of these patients with more advanced tumors undergo neoadjuvant treatment before surgery to try to downstage the tumor. And it gives us an opportunity to biopsy patients before treatment, on treatment, and to also get samples from patients who fail to respond at surgery, so that we can not only, so this gives us a couple of things. Firstly, this is standardized treatment. These patients are all receiving the same drug. We get longitudinal samples, so we can study ecosystems in flux in response to change, as I started out talking about. The other useful thing is that the presence or absence of tumor cells at the end here, known as pathological response, is actually a really good short-term clinical endpoint. People who destroy their cancer through this short burst of chemotherapy generally have very good long-term outcomes. And so this is ongoing. I don't have data to show you about it yet, but I hope it kind of motivates some of you, particularly the clinicians in the audience, to think about how you might build some of these kind of collection protocols into your studies. And I'm really happy to help people if they want to think about how to do this. Okay, I'm going to finish up there. I want to acknowledge the people who've done this work. This is a really fantastic team effort. And acknowledge many of these people. Our collaborators have been wonderful, particularly our clinical leads, Elgene, Munn, and Sandra, our colleagues in the Garvin Weissman Center for Clinical Genomics, funding agencies. And I'll finish there and take questions. Thank you.
Great. Thanks so much, Alex, for a tour de force. That was a really wonderful talk and super exciting work. We have a few minutes for questions. Still a lot of people hanging around. So anything in the chat? Not yet. So I'll just kick off mainly. Yeah, I was actually kind of a bit in your last part of your talk, a bit preempted. A question that I had coming through is the degree to which this approach is likely to reclassify the disease. It's a little surprising, I guess, that the lack of correlation between the your findings around breast cancer ecotypes and the kind of established known genotypes with that obvious clear basal exception in basal cancer. What about other potential factors or patient factors to integrate. So if you look, for example, at germline DNA of patients, I'm just thinking of all the microbiome data, for example, from patients, how these sort of patient intrinsic factors might ultimately determine the endpoint ecotype, and where this all kind of ends up in terms of a sort of grand unifying theory of breast cancer staging.
Yeah, it's a great question, Mark. And obviously there's a ton of work to do to make sense of this. This is really, it's still a pretty qualitative science, to be honest with you. We don't know whether there are 7 ecotypes or 12 ecotypes. These things are sensitive to the way you do the study. And so there's a lot to learn there. But I think your point about what, and this is really the biggest question for me, is what initiates these ecotypes? What kind of coordinates the assembly of these particular cell types into a given tumor? And the obvious first answer is cell intrinsic features of the cancer cells, right? Either their genome or their epigenome sets up a milieu that then kind of signals and coordinates and establishes these communities. And so that's something that thankfully is quite testable. The beauty of these big cohorts like Metabrick, where we started, is that they've been studied like crazy. So they've got very deep genomics, they've got epigenomics, they've presumably got germline data as well. And so that's really one of the next steps for us in this work, is to ask whether there are particular genomic or epigenomic features that associate with ecotypes that might help us understand, what kind of triggers this, what looks like kind of order out of chaos.
All right, there's some heavy hitters coming at you now. So I'll start with, I assume it's Professor Fox, Stephen Fox. Welcome, Stephen. Wants to know in the chat, how stable are the ecotype profiles with regards to the known disease cellular heterogeneity that you see histologically? So I guess how stable are the ecotype profiles across time, do you think? Any evidence of that so far?
Oh, okay, yeah. So there might be 2 questions there. One is stability across time. We don't know, and one of the caveats here as well is that we have, every study like this takes a small sample of the tumor and uses that as our representative sample. And of course, so we'll be very sensitive to sampling bias, and that's a concern. We don't know anything about really the relation to pathology, and I think that's a really interesting point. That we'd be very interested to look into the association between ecotypes and pathological annotation. We haven't done that at all yet, but I imagine these cohorts, such as Metabrick, will have quite a bit of data along those lines. And finally, stability over time. Again, we don't know. It's a pretty difficult one to answer, at least in humans, but I guess our sequential sampling strategy that we're using in the neoadjuvant cohort will be interesting, though, of course, the second time point will be altered by the perturbation of drug treatment. So plenty to figure out, not necessarily always a clean way to get to the bottom of it.
Next question is from Mitch Lawrence. I presume it's from Mitch because Renea previously was dialing in.
I use Mitch's link, Mark.
I don't know if Mitch is like just sort of switching. I don't know if Mitch is like switching rapidly between genders.
Actually Mitch's question. My apologies.
Do you have thoughts on how we can, Alex? Do you have thoughts on how we can model the complex cell-cell interactions you observe?
Yeah, Alex, just to add to that, I think because it was kind of inspiring to see all of those complex interactions, but kind of depressing as well from, people that use patient-derived models to think how we could ever try to recapitulate some of these important interactions in models to try and study those interactions.
Oh, I see what you mean, Mitch. Yeah, it's a great question. And, I think it's something we need to do. So that is something we're doing a bit of work on in the lab, is kind of comparative biology between human disease and model systems. So we've got a series of immunocompetent mouse models of breast cancer, and we've done some pretty deep analysis of them, and we're currently doing that just to try to understand exactly which of these kind of features are conserved and which of them are not. Maybe to alleviate your stress a little bit, we've done a bit of work on breast PDX models as well, which I know you do amazing work with. And certainly, obviously, the immune system is completely different, but the stromal biology is quite well conserved. And one of the things we're interested to do is for all these patients where we have single cell data, we've also xenografted hundreds of them into mice, and we have xenograft models from about 70. So one of the sub-projects we're spinning out now is to do a single cell study on the patient-matched PDXs, which might help us get at some of the questions from earlier as well, to see whether unique stromal features in the patient are conserved in the PDX model. And so that's still a work in progress. We don't have enough cases yet to go from a kind of just a observational study to something harder.
Okay, Ross Dickens doesn't trust me to ask his question, so I'll throw to you, Ross, to ask directly.
That's right. G'day, Alex. Cut out the middleman. Really nice talk, mate. Well done. So a couple of questions about the T cells. So this exhausted TFH set that you see, do you think, so is there a clonotype there that is related to the guys that are still not, that are kind of active and effective, and then some of them are transitioning into the exhausted state? And how also does that relate to the Tregs? Are they all of a similar clonotype?
Yeah, really great question. We can't answer that directly, mostly because we just don't have enough of those cells. We've kind of extracted them from data sets where we've captured every cell type. And to do it properly, we'd have to go back and enrich for them and really smash out thousands of those cells per patient. Because, even in a cancer where you get massive clonal expansion, that might still only be 3 or 5% of the total T cells. So we just can't see that yet. But I think it's a super interesting question. Yeah. Do you see the Tregs have to be there in order for you to see exhausted TFH? Like if you look at individual patients, are they always co-occurring? Great question. We don't know yet. And again, part of the reason is we haven't had enough cases, but in the bigger Atlas cohort where we've got now about 250 cases, that would be something we can hopefully start to get at. And one of the reasons we're scaling like that is so that we can, look for, to identify when this cell is present, what other cells are present or absent kind of questions. Very cool. 260, that's awesome. Well done. That's nuts. Yeah.
Great. All right. I guess maybe just finally a little bit of hand waving, Alex. I mean, where do you see the application of this technology going? Like how might this ever move into a diagnostic setting, do you think? I mean, if you're going to completely reclassify breast cancer using this approach, then and that reclassification is impactful in terms of determining prognosis and particularly selecting patient for treatment A versus treatment B, and is this, I mean, it's a complete hand waving question, but what is the ultimate sort of clinical translatability of this?
Yeah, I mean, it's a great question, and we just don't know. And we're not even, that's almost 2 steps ahead, which is we haven't even got the evidence that this is going to matter at all yet. So really the first experiments we want to do is we need to validate these findings. We need to show that it's at least predictive of response to therapy, which is where it becomes valuable. And we just haven't done that yet. Having said that, let's say we leap that hurdle. Ecotyping, as we've shown, can be done from a bulk RNA-seq sample, and that is something that you could implement clinically. So it's possible that one implementation would be quite simple, which is bulk RNA sequencing. Really what we brought is just kind of the Rosetta Stone. By having these really detailed, clean signatures, that lets you kind of unlock the bulk sequencing and do your cell type predictions. You can, of course, you can imagine other scenarios, some of which we're testing, where you pull out kind of, you only need a handful of markers to identify these ecotypes. That's another possibility, which you could imagine becoming a test that could feasibly work in a hospital, but that's all yet to be shown.
Awesome. All right. Well, we're actually out of time. So thanks for all the questioners, and particularly to Alex for his presentation. Again, it's a real privilege to have you talk to us today, Alex, and thanks so much for making the time for that. And also for the attendees who've dialed in this afternoon, thank you for coming along. So our next seminar is going to be in about a month and a half, on the 23rd of September. It's going to be, I anticipate, another breast cancer talk from Cara Britt, and we'll look forward to circulating the details closer to the date. And don't forget to keep up with upcoming seminars and other information about MPCCC via the website, monashpartnersccc.org, and also subscribing to the newsletter and following us on socials. Awesome. Thanks, Alex, again, and great to see you, and everyone have a great weekend.
Great to see you all. Thanks for coming along.
Very good, mate.